children. Now, I hope all of you are following all these online classes very sincerely, honestly, because all these classes won't be repeated back again for you. So, get ready for the class now. Now, already we have been discussing about this cell organelles. Cell is the structural and functional unit of each and every living organism. Each and every living organism. In your 8th class, you have studied only 3 main parts in it, whereas when you come to 9th class, you have different cell organelles which are floating in cytoplasm. Now let us see what are the cell organelles once again. So first one is cell membrane, second one is cell wall, third one is nucleus, fourth one is cytoplasm, fifth one is mitochondria, sixth one is plastids, next one is ribosome, endoplasmic reticulum, Golgi apparatus, lysosome, vacuole. These are the important cell organelles and each one has its own crucial role in the functioning of the cell. Now listen here, we have already finished about cell membrane. We have covered even cell wall in the previous videos and now we are working with nucleus. As already you know it in your 8th class in the year 1831. Which year ma? 1831. With an experimental material called orchid. Orchid leaves. Mr. Robert Brown. Who is the scientist? Mr. Robert Brown. So he is a scientist who focused on the nucleus present in the cell. Nucleus present in the cell. So, what we have discussed about nucleus is it determines the characteristic feature of the organism. Okay, because it is composed of genetic material in which the characters will be passed from one generation to another generation. For your better knowledge, we can say it as DNA. Deoxyribonucleic acid. Okay, ma? All of you got my point? So, it is composed of what? DNA. The genetic material is present in nucleus. So, as the genetic material is present in the nucleus, it is defining what? It is determining the characteristic feature of the organism. Second, it is the master of the cell. It can be called as manager of the cell, leader of the cell. On a whole, we can say it as cell's control room. What it is called, ma? Cell's control room. So, because it controls all the functions of the cell. It controls all the functions of the cell. So, it controls and regulates all the other cell organs. Now, on a whole, you have seen that cell is composed of so many cell organelles. The nucleus is the master. This nucleus will control all the cell organelles present in the cell. All the cell organelles present in the cell. Now when you see the structure, now it contains what information? Genetic information. That means it has DNA and it involves in cell division. You all know that each and every cell has to be divided. 1 divides into 2, 2 divides into 4, 4 divides into 8, 8 will divide into 16, 16 will divide into 32, 32 will divide into 64. Like that, the division goes on for an organism, multicellular organism to form. So, this needs what division? Cell division. That cell division will be undergone with the help of nucleus. First of all, the nucleus will divide. So, we can say that this nucleus is a cell organelle where the cell division occurs. Where the cell division occurs. Okay? So, it involves in what? Cell division. So, first of all, it determines the character of the organism. It regulates and controls the whole functioning of the cell. That is why we have nicknamed it as cell's control room. Okay, and it carries genetic information and it involves in the process called cell division. Cell division. Okay, and this is the structure of the nucleus. What is this? Structure of the nucleus. So this is the genetic material. Here you can find chromosomes like this. In 10th class you will have more focus on this mark. You will have genetic material and the membrane surrounding this nucleus is called as nuclear membrane. As you remember, the cell surrounding is called as what? Cell membrane. Cell is surrounded by membrane called cell membrane and the nucleus is surrounded by nuclear membrane. Nucleus is surrounded by nuclear membrane. And there is a dark staining body in the midst of the cell, that is nucleus. It is called as nucleolus. Okay? In the midst of the cell, it is nucleus. In the midst of the It is nucleolus. What is that? Nucleolus. I hope all of you are following the pronunciations properly. Right? So, not only this, you will have, this is called nuclear membrane, and the dark staining body present inside the nucleus is nucleolus. And you have regular gaps like this. These are called as nuclear pores. Nuclear pores. And the gel present inside the nucleus is called as nucleoplasm. Nucleoplasm. So this is the structure of nucleus. So clearly you can find the structure of cell in this. Okay, same like cell. Cell is surrounded by cell membrane. Nucleus is surrounded by nuclear membrane. Cell is having a jelly called cytoplasm. Nucleus is having a jelly called nucleoplasm. Okay, understand? Now, cell is having different cell organelles in it. Here, this nucleus is having genetic material in it. Genetic material in it. It is composed of DNA, it is composed of chromosomes. Later on in your further classes, you will know that. Right? And it is having a dark staining body called nucleolus. What is that? Nucleolus. The together whole this structure is called as a nucleus, and the gel present inside the nucleus is nucleoplasm. And for your convenience, what I would like to say you is, few cells are having nucleus, few cells are not having nucleus. Already I have mentioned this for you in 8th class. Few cells are having nucleus, few cells are not having nucleus. The cells with the nucleus are called as eukaryotic cells. The cells without nucleus are called as prokaryotic cell. Understand now? How many types of cells are there? 2 types of cells. Few are prokaryotic, few are eukaryotic. What do you mean by prokaryotic? What do you mean by eukaryotic? Eukaryotic cells means the cells which possess a nucleus. The cells which does not possess a nucleus is called as what? Prokaryotic. What is that called? Prokaryotic. So, basing on the presence of nucleus, we are dividing the cells into 2 types. In the next classes, we will find out what are the differences between eukaryotic and prokaryotic cells. Before that, I would like to say you what is the way you can find nucleus in the cells. Nucleus in the cells. As in the previous classes, we have seen how to observe cell membrane. We have done an activity called how to get cell membrane. And now we are going to see how we are going to observe nucleus for every experiment. So what did I mention here? Lab activity. That means this activity should be done in the lab. So for lab activity, the 5 steps what you have to mention very clearly is, first of all you have to write aim. Second, you have to write apparatus. That can also be termed as materials required. Third one is procedure. Fourth one is observation. Fifth one is inference. Take it granted, even you go to 10th class, even you go to 9th class or any other class, when you are writing an experiment, when you are writing a lab activity, these 5 subheadings are very important. First one is aim, second one is materials required, third one is procedure, fourth one is observation, and the fifth one is inference, result, conclusion. Anything else you can write. Inference, result, conclusion is one and the same. Any term you can use. Now let us start. First, what is our aim is to observe nucleus in the cheek cells. To observe nucleus in which cells? Cheek cells. All of you are having chubby cheeks, no? So, you are going to observe, all of you are going to observe nucleus in which cells, ma? Cheek cells. The experimental material, what we are taking here is our cheek. Our cheek. Right? So, why only cheek means? Why can't we take tongue? What happens if we take tongue? What happens if we take tongue? Now, early morning you will brush your teeth. You will clean your tongue. Why? Why? To remove microbes present in this mouth, in this buccal cavity. Because it gives you a bad smell. Why? Because bacteria called azobacter will be settled on your tongue and on your teeth. So if you take a scraping from your tongue, you will be having what? Bacteria. You can observe bacteria. But we want to observe what we are taking is, we are selecting the experimental material called cheek cells. Now, anyone will allow us to cut the cheek? If I give you a knife and if I ask you to cut the cheek, will you? No, you won't accept. Then how to get a cell from the cheek means, for that what we are going to do is, we are going to select the scrape present in the inner lining of the cheek, not this side, back. You got my point, ma? Inner lining of the mouth you are going to take. On that lining what will be present? Cheek cells will be present. Cheek cells will be present, right? Now let us start. What are the materials required to do this experiment is, first of all, we need a toothpick or ice cream spoon or a spatula. For your convenience, you take ice cream spoon. Okay, anything else you can take. It should not be very hard. It should not damage your lining of your mouth. Lining of your cheek should not be damaged. So for that, what you are going to do is, you are going to take a spatula, you are going to take ice cream spoon, or you are going to take a toothpick, anything for your convenience. Just to collect the scraping from the inner lining of your cheek. Okay, and next you have to take a glass slide. Already you have seen it in your 8th class. There you have observed the slides which are used under microscope, that is nothing but glass slide. Okay, so you have to observe glass slide, and you have to take a cover slip to cover the experimental material. First of all, we have to take a glass slide. We have to take a cover slip. Okay, and next we have to take a watch glass. We have to take a watch glass. So this is glass slide. This is cover slip. This is watch glass. This is what? Watch glass. And we have to take a needle. We have to take a blotting paper. Blotting paper is nothing but tissue paper. When you go to restaurants and all, they'll give you tissue papers to wipe your hands. Like that, you have to take a blotting paper or a tissue paper or a piece of cloth also you can take, right? So you have to take a needle, you have to take a blotting paper, and you have to take 1% methylene blue. This is very important. This is the stain what we are using. This is called stain. What is this called? Stain. You can use red ink, you can use blue ink, you can use iodine, anything else, but we have to stain the experimental material. So we are using this color called methylene blue. Methylene blue and normal saline. Normal saline. That is nothing but salt water. Next one is glycerin. Next one is what? Glycerin. After the experiment is over, we have to observe it under the microscope, because we can't see the cells directly. We can observe this staining technique only under electron microscope. Okay, there are 3 kinds of microscope: simple microscope, compound microscope, electron microscope. Now we have to take a microscope. Now what we are going to do is, first of all, wash your mouth, rinse your mouth, take an ice cream spoon and just collect the scraping, and keep all the scraping collected on the watch glass. Don't keep it directly on the slide. Don't keep it directly on the slide. Keep it on the watch glass, which is already provided with normal saline. Why to use normal saline means it won't get dry. Right? Now here in the watch glass, we are collecting the scraping of the cheek, and we are just collecting and keeping it on the watch glass. So first of all, we have to wash your mouth. And next we have to collect the inner lining of your cheek with the help of toothpick, ice cream spoon or spatula. So we have selected what? Ice cream spoon. So with the help of ice cream spoon, we have collected the scraping and the inner lining of the mouth. Okay, and we kept it on the watch glass, which contains normal saline. And now we have to collect this material which is collected on the watch glass. We have to transfer it onto the glass slide with the help of needle. That's why we have mentioned needle here. Okay, see, check glass slide. To the glass slide. To the glass slide. Now, what we have to do is transfer it to the glass slide. Now, we have taken 1% methylene blue. 1% methylene blue. Now, place 2 or 3 drops of methylene blue on it. On the material what we have selected. Okay? On the material what we have selected, that is cheap. Right? You just drop 1% methylene blue for 2 or 3 drops. Now, we have to wait for 2 minutes. For a couple of minutes, you have to keep the slide like that only without disturbing. Without disturbing. Now, after 2 minutes, take the blotting paper. Take the blotting paper and remove the excess stain from the slide. Now what we have to going to do is wipe off the extra stain with what? Tissue paper or blotting paper. Now put a drop of glycerin on it. Put a drop of glycerin on it. After keeping glycerin on it, cover it with the cover slip. Cover it with the cover slip. Cover it with the cover slip. And after covering it with cover slip, we have to observe it under the microscope. After observing it under the microscope, you can observe that there are cells like this observed with a dark staining body. With a dark staining body in mid, one dark staining body is present. This is called as nucleus. This is called as what? Nucleus. This is called as nucleus. So by this we can understand that. So this is the procedure what you have to follow. And next in the observation we can see, when you observe it under the microscope, you can clearly observe that dark staining body is present in the midst of each and every cell observed under microscope. That dark staining body is nothing but nucleus. What is that? Nucleus. Thus it is proved that nucleus is present inside the Chi cells. Nucleus is present inside the Chi cells. So this is the way how you can observe nucleus in the Chi cells. So now the cells which are having nucleus is called as eukaryotic. The cells without nucleus are called as prokaryotic. There will be some major differences between these two. In the next videos, we'll have this class